Have you ever paused to wonder how we truly make sense of the dizzying, incredible diversity of life on Earth? I mean, from the tiniest bacteria you can barely see, all the way up to the largest whale swimming in our oceans. How do scientists actually organize all of that?
It's a foundational question, isn't it? It's
Uh-huh.
not just about giving things names. It's about understanding the deep relationships, the evolutionary pathways, and how every form of life fits into a grand, interconnected system.
That's precisely what this deep dive is all about: biological classification. We're going to explore how we've tried to put living organisms into neat little boxes over centuries, why those boxes have continually shifted as our knowledge grew, and what fascinating insights these systems reveal about life itself.
Hmm.
Our mission today is to unpack the evolution of these ideas, focusing especially on the powerful Five Kingdom system, and to highlight some surprising facts and characteristics you're definitely going to remember.
And we've drawn from a range of expert sources on biology and classification systems to guide us through this intricate map of life.
Okay, let's unpack this journey, because the way we classify life has truly evolved right alongside our understanding. Think back to the dawn of civilization. Classification wasn't some grand scientific pursuit then. It was instinctive, right? Driven by immediate human needs.
Absolutely. Yeah.
You classified plants and animals because you needed to eat them, find shelter, or make clothing. It was about survival, basically.
Absolutely. A practical, almost intuitive necessity. But then we start to see the first glimmer of a more structured approach with someone like Aristotle. He was one of the earliest to attempt a more, let's say, scientific basis.
Okay.
For plants, he used simple morphological characters, just what they look like, to divide them into trees, shrubs, and herbs.
Hmm.
Pretty straightforward.
Right. And for animals, he had two very simple groups: those that had red blood and those that did not. So your primary lab equipment was your eyes, and maybe a very brave assistant. Very basic, but it was a start.
Exactly, a start. Fast forward quite a bit, and we arrive at Linnaeus's time. This is where the two kingdom system really took hold, dividing all life into just Plantae and Animalia. On the surface, it seemed intuitive and easy to grasp.
Yeah, that makes sense. Plants are plants, animals are animals. Seems pretty clear. But here's where it gets really interesting, because this system, despite its initial appeal, had some significant shortcomings. What were the biggest oversights? Where did it really fall short?
Well, for all its revolutionary impact, the Linnaean system did not distinguish between eukaryotes and prokaryotes. That's a huge oversight, considering how fundamentally different their cell structures are.
Okay, can you just quickly remind us what those terms mean?
Sure. Prokaryotes are organisms whose cells lack a true nucleus and other membrane-bound organelles, like bacteria. Eukaryotes, like us, plants, fungi, they do have cells with a nucleus and all those internal structures.
Got it. So no prokaryote to karyote distinction. What else?
It also did not separate unicellular and multicellular organisms. Think about it. A single-celled bacterium and a massive elephant would be lumped into just two kingdoms under that old system. Doesn't quite capture the complexity, does it?
Not really, huh?
And crucially, it failed to differentiate between photosynthetic organisms, like green algae, which make their own food, and non-photosynthetic organisms, like fungi, which don't.
Ah, so fungi were just stuck in the plant kingdom. Why?
Well, basically because they didn't move like animals and often had cell walls. There's a very superficial grouping. They weren't making their own food, but they ended up with the plants.
So as you can imagine, a large number of organisms simply did not fall neatly into either the plant or animal category.
Exactly. This inadequacy made it clear that classification systems needed to consider more complex characteristics beyond just, what something looked like on the outside. Scientists needed to look deeper.
At things like,
Things like cell structure, that prokaryote eukaryote thing, the nature of the cell wall, how they got their nutrition, their habitat, reproduction methods, and vitally evolutionary relationships, their phylogeny.
and that brings us to the game changer, R.H. Whitaker's Five Kingdom classification, proposed in 1969. This is where things get truly refined and fascinating because it addresses so many of those earlier shortcomings.
Whitaker proposed the pivotal five-kingdom classification, and those kingdoms are Monera, Protista, Fungi, Plantae, and Animalia. These are the categories that still form a fundamental understanding of life's organization today for many biologists.
So what made Whitaker's system so much more robust? It wasn't just about adding more categories, was it?
Not at all. The criteria Whitaker used were significantly more detailed and insightful. He looked at cell structure, body organization, like unicellular versus multicellular, mode of nutrition, reproduction, and crucially, phylogenetic relationships, those evolutionary connections.
Right, that multifaceted approach.
Yes, it allowed for much finer distinctions and, well, helped us understand life's diversity more accurately.
And that's where the data really comes into play, because when you look at these characteristics across the 5 kingdoms side by side, you can see how truly different they are. Let's really dig into that, focusing on the key data points for each. This is the core of it.
Okay, let's start with Monera. This kingdom is absolutely unique because it is the only one that is prokaryotic. Remember that. Monera is the sole prokaryotic kingdom. No true nucleus.
Prokaryotic, got it. It's the only one.
Its cell wall is also unique. It's made of a non-cellulose material, a combination of polysaccharide and amino acid. Very different from plants.
Non-cellulose wall, okay.
And a key detail linked to being prokaryotic. The nuclear membrane is absent in Monera. The body organization is always cellular, meaning it's single-celled.
Single-celled, prokaryotic, no nuclear membrane, non-cellulose wall. What about food?
For nutrition, Monera is incredibly diverse. It can be autotrophic, making its own food through chemosynthesis or photosynthesis, and it can be heterotrophic, acting as a saprophyte, feeding on dead stuff, or a parasite living off a host. Huge range.
So Monera stands out immediately. The only prokaryotic group, no nuclear membrane, unique cell wall, super diverse nutrition.
Got it. What about Protista?
Protista is our first eukaryotic kingdom in this system. So unlike Monera, it does have a nuclear membrane, which is present.
Okay, eukaryotic. Big difference right there.
Yeah. For their cell wall, it's a bit mixed. A cell wall is present in some, but not all. Like Monera, their body organization is cellular. They're primarily single-celled eukaryotes.
Single-celled eukaryotes.
Yeah.
And nutrition.
Their nutrition is also mixed, capable of being both autotrophic, photosynthetic, and heterotrophic. So Protista really represents this sort of catch-all kingdom for the early single-celled eukaryotes before things got more complex. It bridges the gap in some ways.
Okay, so Protista, single-celled eukaryotes, nuclear membrane present, some have cell walls, mixed nutrition. What about fungi?
Fungi are also eukaryotic, so nuclear membrane present. And a critical detail is their cell wall. It's present, but it's made with chitin. This is distinct from plant cell walls, a major data point for differentiation. Remember, chitin.
Right.
Not cellulose like plants.
Exactly. Their body organization is typically multicellular or loose tissue, though some, like yeasts, are unicellular. But here's a crucial characteristic. Fungi are strictly heterotrophic. They get their food from other sources, often as saprophytes, breaking down dead matter, or as parasites. They never make their own food.
So fungi, eukaryotic, mostly multicellular. That chitin cell wall is a big differentiator, and they're purely heterotrophic.
Got it. Now to the familiar Plantae.
Plantae are, of course, eukaryotic, nuclear membrane present. Their cell wall is present, and importantly, it's made of cellulose. That's the key plant cell wall component. Cellulose.
Cellulose for plants, chitin for fungi.
Okay. Body organization is more complex, forming tissue or organs, and their mode of nutrition. Almost exclusively autotrophic through photosynthesis. They are the primary producers for much of life on Earth.
So plants, eukaryotic, cellulose cell wall, tissue organ structure, and the primary food makers. And finally, Animalia. Animalia are also eukaryotic, nuclear membrane present. Here's a key distinction. Their cell wall is absent. That's a huge differentiating factor from plants and fungi. No cell wall.
No cell wall at all. That's big.
It is. Body organization is the most complex, ranging from tissue to organ systems. And their nutrition is entirely heterotrophic, often allozoic, meaning they ingest their food.
So when you lay it out like that, the differences become really clear. Monera jumps out as the only prokaryotic one. Plantae and fungi, both eukaryotic with cell walls, but totally different wall materials, cellulose versus chitin, and animals. Eukaryotic, complex bodies, but no cell wall. These are the core data points that really define each kingdom.
Exactly. This side-by-side comparison based on multiple criteria is essential because it shows why Whitaker's system was such a leap forward. Earlier systems, which often lumped organisms based only on one or two traits, like having a cell wall, made some truly strange groupings.
Yeah, like placing prokaryotic bacteria and blue-green algae cyanobacteria right alongside eukaryotic groups under plants. That just doesn't make sense when you consider the fundamental difference in cell structure, prokaryotic versus eukaryotic.
Or grouping unicellular organisms, like Chlamydomonas, with multicellular ones, like Spirogyra, all under algae within plants. They are fundamentally different in body organization. One's a single cell, the other's multicellular.
Right.
And probably the biggest issue, as we mentioned, was failing to differentiate heterotrophic fungi. with their distinct chitin walls from autotrophic green plants, which have cellulosic walls. Just lumping them together because they weren't animals wasn't good enough.
So how did Whitaker's new criteria actually solve these issues? What was the breakthrough?
By focusing on those multiple characteristics, cell structure, prokaryote, eukaryote, nutrition, autotroph, heterotroph, cell wall composition, cellulosic and absent, body organization, Whitaker could finally give fungi their own separate kingdom.
Finally, fungi get their own kingdom.
Yes. This meant they were no longer miscategorized as plants. All prokaryotic organisms were united under Kingdom Monera, recognizing their shared fundamental cellular organization. And all unicellular eukaryotic organisms, regardless of their other traits, were placed together in Kingdom Protista.
That's the aha moment. So this meant organisms like Chlamydomonas and Chlorella, which are single-celled green algae, were earlier in plants, but under Whitaker.
Under Whitaker, they moved to Protista. They were pulled out of Plantae and grouped with other single-celled eukaryotes like Paramecium and amoeba, which interestingly used to be classified in the animal kingdom because they moved and ate.
Wow, so things really got shuffled around.
They did, because the
Yeah.
criteria for classification changed. It wasn't just about one simple trait anymore. It was about a combination of fundamental biological characteristics.
That makes so much more sense.
Precisely. And it's important to remember that classification is always evolving. Science doesn't stand still. For instance, you might hear about the three-domain system proposed by Carl Woese, which actually divides Kingdom Monera into two domains, bacteria and archaea, with eukarya as the third. This leads to what some call a six-kingdom classification. Our understanding continues to deepen, especially with genetics.
Always refining. Okay, now that we've seen how Whitaker built a more logical framework, let's peel back the layers and discover some truly surprising and often unseen details about the inhabitants of each of these five kingdoms. Starting with Kingdom Monera, the bacteria truly the abundant masters of our world.
You can't overstate their numbers. Bacteria are the most abundant microorganisms on Earth. Our sources tell us that hundreds of bacteria are present in a handful of soil. Think about that. Just a handful of soil holds hundreds, maybe thousands or millions of individual bacteria.
That's incredible.
They live almost everywhere, thriving in extreme habitats like scorching hot springs, arid deserts, icy snow, and the crushing depths of the oceans where you'd think nothing could survive.
They're everywhere. And they come in distinct shapes, too, which helps us classify them further, right? You mentioned shapes earlier.
Yes, bacteria are grouped into four primary categories based on their shape. You have the spherical coccus, plural cocci, which are often found in clusters or chains. And there's the rod-shaped bacillus, plural milzilli, which are quite common.
Coccus and bacillus, round and rod.
You also have the comma-shaped vibrium, plural vibrio, which look like tiny curved rods, like the one that causes cholera. And finally, the spiral spirillum, which, as the name suggests, are helical or corkscrew-shaped.
So you've got your round ones, your rod-shaped ones, your comma-shaped ones, and your spirals. Coccus, bacillus, vibrium, spirillum, those are the four main shapes to remember.
Exactly. Key data points for identification. And metabolically. They show the most extensive metabolic diversity of any kingdom. We already touched on this: autotrophic, photosynthetic or chemosynthetic, and heterotrophic, the vast majority, including decomposers.
This vast metabolic diversity, does it tell us something about early life?
It likely does. It suggests they were incredibly resourceful and adaptable from very early on, able to exploit almost any energy source available in the harsh conditions of early Earth.
And you mentioned archaebacteria specifically?
Yes, archaebacteria are fascinating. They're a group within Monera known for living in extreme environments. We're talking about halophiles in extremely salty areas like the Dead Sea, thermoacidophiles thriving and boiling, acidic hot springs, and methanogens in oxygen-poor marshy environments.
Methanogens. They make methane.
That's right. Methanogens are crucial. They're found in the gut of ruminant animals like cows and buffalo, and they're responsible for producing methane gas, which we know is biogas from the animal's dung.
So they have a direct impact on our environment and even potential energy sources. What about eubacteria, the true bacteria?
They have a huge impact, too. Cyanobacteria, or blue-green algae, are eubacteria. They have chlorophyll A, similar to plants, making them photosynthetic oxygen producers. Crucially, many can also fix atmospheric nitrogen, like nostoc and anabina, enriching soils.
And the heterotrophic ones.
Heterotrophic bacteria are the most abundant type. They're the great decomposers, breaking down dead organisms and waste. But they also play vital roles for us, making curds from milk, producing antibiotics like streptomycin, fixing nitrogen in the roots of legume plants. But of course,
There's a downside.
yes, some are pathogens, causing serious human diseases like cholera, typhoid, tetanus, citrus canker in plants. The list goes on.
And you mentioned one more really weird group in Monera, mycoplasma.
Ah, yes, mycoplasma, truly unique. They completely lack a cell wall. This is a major defining feature, very unusual for bacteria. They are also the smallest living cells known, incredibly tiny. And remarkably, they can survive without oxygen.
Smallest living cells, no cell wall. Wow, are they harmful?
Many are pathogenic in animals and plants, yes. Their lack of a cell wall makes them resistant to certain antibiotics like penicillin that target cell wall synthesis.
Fascinating. Okay, moving on to kingdom Protista, the single-celled eukaryotic wonders. You said they're primarily aquatic and link the other kingdoms.
That's right. They're kind of the ancestors or early forms leading towards plants, animals, and fungi. Among them are the chrysophytes, which include diatoms and golden algae. Diatoms are incredible.
Why is that?
Their cell walls are like little glass boxes embedded with silica, making them practically indestructible. Over billions of years, these silica shells accumulate on the ocean floor, forming vast deposits of diatomaceous earth.
And we use that.
We do. It's used in polishing, filtration of oils and syrups. It's gritty stuff. But here's the massive ecological impact. Diatoms are the chief producers in the oceans.
Chief producers. What does that mean exactly? More than algae.
It means they are responsible for a huge chunk of the photosynthesis happening in the marine environment. They form the very base of the marine food web. Think about that these microscopic single cells are fundamental to supporting nearly all other life in the oceans. A massive impact.
That's staggering for something microscopic. And then there are the dinoflagellates, known for something maybe less positive.
Yes, the red tides. Some dinoflagellates, like gonilacs, contain red pigments. When conditions are right, they can multiply incredibly rapidly, turning vast areas of the sea red.
That sounds dramatic. Is it dangerous?
It can be. Many of these red tide dinoflagellates release toxins that are potent enough to kill fish and other marine animals that consume them or are exposed.
Okay. What about slime molds? The name is evocative.
Yes. Slime molds are saprophytic protists, meaning they feed on decaying organic matter. They have a fascinating life cycle. Under suitable conditions, they form an aggregation called a plasmodium, which is essentially a multinucleate mass of protoplasm without cell walls. This plasmodium can grow and creep, sometimes spreading over several feet, engulfing food particles like a giant amoeba.
Like something out of a sci-fi movie? And
Right, ma'am.
protozoans. You mentioned they're like primitive relatives of animals.
Yes, they heterotrophic protists, often predatory or parasitic. There are several groups like amoeboids, flagellates, ciliates. But one group, the sporozoans, has a particularly significant impact on humans.
Which one is that?
This group includes organisms that have an infectious, spore-like stage in their life cycle. The most notorious example is Plasmodium.
Plasmodium. That sounds familiar.
It should. Plasmodium is the parasite that causes malaria. It requires both mosquitoes and humans to complete its life cycle. And malaria, as a disease, has had and continues to have a staggering effect on human population across the globe, particularly in tropical regions. A microscopic protist with enormous consequences.
A huge global health issue stemming from a tiny sporozoan. Okay. Next, kingdom fungi. The heterotrophic recyclers. You often see them in warm, humid places, hence refrigerating food.
Precisely. They thrive in warmth and moisture, which is why bread gets moldy quickly on the counter, but lasts longer in the fridge. They're cosmopolitan, found everywhere.
And their structure, you mentioned hyphae and mycelium.
Yes. Except for yeasts, which are unicellular, fungi typically have bodies consisting of long, slender, thread-like structures called hyphae. These hyphae weave together to form a network called mycelium, which is the main body of the fungus, often hidden in the soil or whatever it's growing on.
And that cell wall, again, what was it made of?
Right, crucial point. Fungal cell walls are uniquely made of chitin and polysaccharides. Remember, chitin, not cellulose. It's the same tough material found in the exoskeletons of insects.
Chitin. Got it. And their nutrition, strictly heterotrophic.
Correct. Mostly saprophytes, absorbing soluble organic matter from dead substrates. Some are parasites living on plants or animals, and others are symbionts, living in mutually beneficial relationships like lichens, which are fungi living with algae, or mycorrhiza, fungi associated with plant roots.
And they're useful, too, beyond just breaking things down. We use them.
Absolutely. Yeast, a unicellular fungus, is vital for fermentation, making bread rise and brewing beer and wine. And many fungi produce antibiotics. The famous example is penicillium, the source of penicillin, Some mushrooms are edible, though many others are poisonous.
Right, important distinction there. Now, for the kingdoms perhaps most familiar to us, Kingdom Plantae and Kingdom Animalia.
Okay, Plantae. These are eukaryotic, chlorophyll-containing organisms. Key feature. Their cells have a strong cell wall made primarily of cellulose. That's the defining structural component.
Cellulose walls, eukaryotic, make their own food? Seems straightforward.
Mostly, they are primarily autotrophic, using photosynthesis. However, nature loves exceptions. There are some fascinating partially heterotrophic plants.
Plants that eat.
Well, sort of supplement their diet. Think of insectivorous plants like the bladderwort and Venus flytrap. They photosynthesize, but they also trap and digest insects to get extra nutrients, especially nitrogen, often because they live in nutrient-poor soils.
Clever adaptation. Any others?
There's also the parasitic plant Cuscuta, or daughter. It lacks chlorophyll and wraps around a host plant, tapping into its vascular tissue to steal water and nutrients. So yes, most plants are autotrophs, but there are these surprising exceptions.
Adds a little twist. And Animalia, our own kingdom.
Characterized by heterotrophic, eukaryotic, multicellular organisms that distinctly lack cell walls. That absence of a rigid cell wall is a key feature.
No cell walls. We rely on skeletons or other structures for support instead.
Exactly. Animals digest their food in an internal cavity and store food reserves, typically as glycogen or fat. Their nutrition is holozoic, meaning they ingest complex food and digest it internally. They generally show definite growth patterns, develop into adults with a definite shape and size, and most are capable of locomotion, moving around. They also have more complex sensory and neuromotor mechanisms compared to other kingdoms.
Okay, so that covers the 5 kingdoms, but what about the organisms that Whitaker's system doesn't classify? These outliers. Viruses, viroids, prions, and lichens. Why aren't they in the 5 kingdoms?
That's a great question. They aren't mentioned in Whitaker's 5-kingdom system, primarily because they don't fit the fundamental criteria, particularly the definition of being cellular life in the way the kingdoms are defined. Viruses, viroids, and prions are non-cellular. Lichens are composite organisms. They don't neatly fit into any single kingdom box.
Let's start with viruses. They're often called borderline life forms. The name itself means venom or poisonous fluid.
That's right. It reflects early observations of infectious agents. Their history is quite interesting. Dmitry Ivanovsky, back in 1892, discovered that the agent causing tobacco mosaic disease could pass through filters that trapped bacteria, so it had to be smaller.
Smaller than bacteria.
Yes. Then Martinus Beijerinck in 1898 demonstrated that this agent could reproduce within host cells and called it contagium vivum fluidum, meaning infectious living fluid. But the real breakthrough in understanding their nature came with Wendell Stanley in 1935.
What did Stanley show?
He showed that viruses could be crystallized. Think about that. You can crystallize them like salt. And these crystals consist largely of proteins. This suggested they were inert, non-living structures outside of a host cell.
So they're like dormant packages of chemicals until they get inside a living cell, then they spring to life, hijacking the cell's machinery to replicate. That's why the debate about whether they're truly living continues.
Precisely. They exist at the edge of life. And here's a crucial genetic material fact about viruses, something really important to remember for differentiating them. Viruses contain genetic material that is either RNA or DNA, but never both.
Okay, say that again.
A virus will have a core of genetic material, but it will be either RNA or DNA. It never possesses both simultaneously, unlike cellular life, which uses both.
So no virus will ever have both RNA and DNA together. That's a fundamental rule for viruses.
It is. Plant viruses, for example, usually have single-stranded RNA. Animal viruses can have either single or double-stranded RNA or double-stranded DNA. Bacteriophages, the viruses that infect bacteria, usually have double-stranded DNA.
And they cause a lot of familiar diseases, right?
Unfortunately, yes. They're responsible for common ailments like the common cold and influenza, but also serious diseases like mumps, smallpox, now eradicated, herpes, and AIDS, which is caused by HIV. In plants, they cause mosaic formation, leaf rolling, yellowing, stunting, lots of damage.
Okay. What about viroids? You said even smaller.
Yes, viroids are even smaller than viruses. Discovered by T.O. Diener in 1971, he found the agent causing potato spindle tuber disease was smaller than any known virus. And here's a key difference. Viroids are essentially free RNA. They consist only of a short strand of RNA. And crucially, they LACK the protein coat that viruses have.
Just naked RNA causing disease.
Exactly. Free RNA, no protein coat. And the RNA has a low molecular weight.
Fascinatingly simple yet infectious. And prions, what are those? They sound different again.
Prions are a whole other really strange category.
Yeah.
They are infectious agents consisting only of abnormally folded proteins, similar in size to viruses, but just protein, no RNA or DNA involved at all.
Infectious proteins, how does that even work?
It's thought that these misfolded prions can induce normally folded proteins in the host, especially in the brain, to also misfold, leading to a chain reaction and disease. They cause fatal neurodegenerative diseases.
Like what?
The most well-known examples are bovine spongiform encephalopathy, BSE, commonly known as mad cow disease in cattle, and its human variant, Creutzfeldt-Jakob disease, CJD. It's quite alarming that just a misfolded protein can be infectious and cause such devastating effects.
Truly mind-bending. Lastly, lichens. You mentioned they're symbiotic. Are they just one organism or?
No, lichens are a classic example of symbiosis, a mutually beneficial relationship between two completely different organisms. Algae and fungi. They live together so intimately that they appear as a single entity.
So who does what in the partnership?
The algal component, called the phycobiont, which is usually green algae or cyanobacteria, is autotrophic and makes food for both partners through photosynthesis. The fungal component, the mycobiont, provides structure, shelter, and absorbs mineral nutrients and water from the environment for the alga.
A perfect partnership.
It really is. They're so closely associated. And here's an important ecological fact about them. Lichens are known to be very good pollution indicators.
How so?
They are very sensitive to air pollution, especially sulfur dioxide. They simply won't grow or will die off in heavily polluted areas. So their presence or absence can tell you a lot about the air quality.
A natural early warning system. So we've journeyed from Aristotle's simple observations of trees and red blood through Linnaeus's basic two-kingdom system and landed in the much more detailed and, frankly, more accurate world of Whitaker's five kingdoms. We saw how deeper understanding of fundamental biology's cell structure, nutrition, genetics, evolutionary relationships forced these classifications to evolve.
In understanding these categories, Monera, Protista, Fungi, Plantae, Animalia, and even the outliers like viruses, isn't just academic. It's a shortcut, really, to being well-informed about the incredible diversity of life on our planet. It helps us see the patterns, the connections, and the unique strategies different organisms employ just to survive and thrive.
This deep dive really highlights that our classification systems are dynamic, always changing as our knowledge grows. So as you go about your day, maybe look at the life around you a bit differently and think about this. What new discoveries, perhaps in genetics with DNA sequencing, or maybe even AI analyzing complex traits, do you think might lead to the next big shift in how we classify life? And how might that change our fundamental understanding of what it truly means to be alive?